Today we will learn about the glycolysis. When we think about the glycolysis, it is also known as a Embden Meyerhof Pathway or EM pathway, presented by myself, Preetham Zuatka, Assistant Professor, Kokan Ganpit, Rahul Dhaka College of Pharmacy and Research Institute, Karjat. When we think about specifically glycolysis, this glycolysis is nothing but oxidation of glucose, but it is defined as sequence of a reaction converting glucose to pyruvate or lactate with the production of ATP. So glycolysis is defined as a sequence of a reaction converting glucose to pyruvate or lactate with the production of ATP. So glycolysis takes place in all cells because the enzyme present in this is nothing but present in a cytosomal fraction of a cell. Glycolysis occurs in presence of oxygen, that is known as aerobic condition, and the product of it is known as a pyruvate, and in absence of oxygen, that is known as an anaerobic condition, that product is known as a lactate. When we think about this end product of this aerobic under the anaerobic condition is oxidized to CO2 and H2O. So that's why it is a sequence of reactions converting glucose to pyruvate and or lactate. So glycolysis is a major pathway for ATP synthesis in a tissue lacking a mitochondria. So we know mitochondria is a powerhouse unit or power unit of our cell. But this is a one other pathway, glycolysis, which is required, a major pathway where there is some cells are there lacking in mitochondria. Say example, erythrocytes and cornea and lens. So glycolysis is a central metabolic pathway, and this central metabolic pathway producing various intermediates product, and these intermediates are utilized for the synthesis of amino acid as well as a fat in the body. Some you there are nine steps are there in the glycolysis. These 9 steps are very important, and out of this, total is a 10 product. So we're having three reactions are irreversible. Three reactions are irreversible in glycolysis. And these reactions are nothing but it is converted again into reversible form in a specific condition. And that is known as a, say example, your body is not having a glucose and your brain is utilize only the glucose energy. So when body does not having intake of glucose, the brain requires the energy and that will fulfill by converting non-carbohydrate product into a carbohydrate product, that is a glucose. And that process, synthesis of glucose, is known as a gluconeogenesis. So in this, these three irreversible steps of glycolysis is converted into, with the help of some enzyme, it converted into reversible, and this reversible glycolysis is one of the part of gluconeogenesis. Now we will concentrate on specifically on glycolysis. So we're having a glycolysis is having 3 steps or 3 distinct phases, and that first is energy investment phase or primitive stage. Second phase is known as a splitting phase, and third phase is known as a energy generation phase. Now we will move towards a first phase, that is energy investment phase. This energy investment phase is a specifically in this reaction glucose is phosphorylated to glucose 6 phosphate in the presence of glucokinase or hexokinase. This you can able to see here, glucose molecule consists of the 6 carbon moiety, and this is an irreversible reaction. First investment phase, energy investment phase, first reaction is irreversible reaction. It in the presence of one enzyme that we know is known as a hexokinase. Now what will happen? This glucose converted to glucose 6 phosphate in the presence of hexokinase. Here the energy is utilized because glucose is been glucose 6 phosphate. So phosphate is coming from the ATP, and this ATP in the presence of magnesium ion, it is converted to ADP. So first reaction is glucose is phosphorylated. Glucose is nothing but a phosphorylated by use of ATP, which is converted into ADP. There is adenosine triphosphate converted into adenosine diphosphate, and one phosphate molecule will bind to the 6 carbon in the presence of hexokinase. And this hexokinase is present in all tissue, almost all tissue it has been present. You can see the changes which is occurring in the 6 carbon here. Now, the next reaction of the energy investment phase is this glucose 6 phosphate isomerization reaction. So it is isomer of the glucose is nothing but fructose. So glucose 6 phosphate can isomerize to form a fructose 6 phosphate. Glucose 6 phosphate isomerize to form a fructose 6 phosphate. We require the one enzyme that is definitely it is isomerase enzyme, because isomerization reaction means what? Isomerase enzyme. And here we having converting glucose 6 phosphate into fructose 6 phosphate. So phospho and hexo sugar. So that's why enzyme name will be phospho hexose, and reaction is isomerization. So that's why the enzyme will be phospho hexose isomerase. This is one of, we can say it is a reversible type of reaction in the presence of magnesium ion, which is nothing but required for this reaction. And what is the changes occurring? You can able to see here. This is nothing but the, it will get shifted to the second carbon, that is nothing but the bonding, and this is nothing but the isomers to each other. So glucose 6 phosphate convert into fructose 6 phosphate in the presence of phosphohexose isomerase, and the reaction is isomerization. Now next reaction, this will be the last reaction of energy investment phase. This is a last reaction of energy investment phase. In three reaction, one was irreversible. This is known as a irreversible reaction again. Here also ATP is utilized. Now fructose 6 phosphate. This is a product of previous reaction. It is irreversible reaction in the presence of phosphofructokinase. In the presence of phosphofructokinase, and this ATP converted into ADP, and magnesium ion is required for this reaction. It will form a fructose 1,6 bisphosphate. So in a previous first reaction, that was a hexokinase enzyme, and here phosphofructokinase enzyme. You can easily notify that fructose 6 phosphate converted into fructose 1,6 bisphosphate in the presence of phosphofructokinase. So kinase enzyme plays an important role is a phosphorylation and dephosphorylation. Here we have phosphorylation, and the enzyme name is phospho, fructo, and the reaction is kinase. So you can easily remember this fructose 6 phosphate convert into fructose 1,6 bisphosphate in the presence of phosphofructokinase, and here this changes you can able to see here. And this is nothing but the, we can say we were completing the three phases of glycolysis. Now we are moving towards the splitting phase. This splitting phase is a specifically we can say the 6 carbon moiety, that is known as a fructose 1,6 bisphosphate, that will get split. Split means what? Divide. Split into 2,3 carbon compound. We can able to see here in this fructose 1,6 phosphate is there. This fructose 1,6 bisphosphate, it will get split. This splitting in the presence of one enzyme known as a aldolase, and that will form by breaking this bond in between the fourth and fifth. It will get break, and that is nothing but it is forming a product known as a 3 carbon compound glyceraldehyde 3 phosphate, and another one is known as a dihydroxyacetone phosphate. These are the 3 carbon compound which are present in the reaction. So splitting phase is a one reaction only, but here the products are two. So splitting phase, in this 6 carbon fructose 1,6 bisphosphate, it will get split into 3 carbon compound. One is known as a glyceraldehyde 3 phosphate, and another is known as a dihydroxyacetone phosphate in the presence of one enzyme known as a aldolase. See what will happen, these two compounds are specifically isomers to each other. These are isomers to each other. So what will happen? The glyceraldehyde 3 phosphate can convert into dihydroxyacetone phosphate, and dihydroxyacetone phosphate can convert into glyceraldehyde phosphate. So you can easily make out the two glyceraldehyde 3 phosphate molecules will form. And this is nothing but a reversible again reaction. Both reactions are reversible. See, I had used a specific color identification in which what red color is defined as a substrate and product. Green color is defined as a specifically enzyme, and for the reversible reaction I had used, it is for the only to understand the and to memorize. So this splitting phase is over with the phosphotriisomerase can convert two molecules of forming a glyceraldehyde 3 phosphate can form a dihydroxyacetone phosphate, and dihydroxyacetone can convert into glyceraldehyde 3 phosphate. Now we are moving towards the third and last phase of the glycolysis. This is known as the energy generation phase. This energy generation phase, in that the first reaction is what? Two molecules of glyceraldehyde 3 phosphate. In splitting phase, we got the two molecules of glyceraldehyde 3 phosphate. These two molecules of glyceraldehyde 3 phosphate will convert into a high energy compound that is known as a 1,3 bisphosphoglycerate. This will convert to form a one high energy compound that is known as a 1,3 bisphosphoglycerate. And this particularly we can say in this reaction what will happen? The NADH, that is nothing but here the NAD plus convert into NADH and H plus. Here, what will happen? Glyceraldehyde 3 phosphate can convert into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. And here, NAD convert into NADH and H plus, and here that a phosphate molecule plays an important role in the ATC, and this nothing but known as oxidative phosphorylation, synthesized by the oxidative phosphorylation. And this is nothing but we can say a first energy compound has been formed, or a first energy in generation phase reaction is there, and this will nothing but the forming a 1,3 bisphosphoglycerate. So what is there in this reaction? Glyceraldehyde 3 phosphate can convert into 1,3 bisphosphoglycerate in the presence of glyceraldehyde 3 phosphate dehydrogenase. Hydrogen has been removed, dehydrogenase, so NAD convert into NADH and H plus. This is nothing but we can able to get in this a 6 ATP. See, electron transport chain mechanism, NADH is passes through the electron transport chain mechanism and forming a total 3 ATP, and here the two molecules have been used, so there will be a 6 ATPs will be forming in this reaction. Now we will move towards the specifically we can say the next reaction of the energy generation phase, reaction number 6. So in this 1,3 bisphosphoglycerate, which is a high energy compound, and that forming a 3 phosphoglycerate. 1,3 bisphosphoglycerate, this compound will form a reversible reaction. It is nothing but in the presence of phosphoglycerate kinase, again kinase enzyme, phosphoglycerate kinase, and this nothing but forming a 3 phosphoglycerate. So what will happen in this reaction? ADP convert into ATP. This is nothing but known as a substrate level phosphorylation. Substrate level phosphorylation means what? Here this substrate level phosphorylation in which was substrate without involvement of ETC, that is electron transport chain mechanism. Without involvement of the ETC, it is forming a ATP energy, and that's why it is known as a substrate level phosphorylation. So in this reaction 1,3 bisphosphoglycerate convert into 3 phosphoglycerate in the presence of phosphoglycerate kinase, and ADP convert into ATP. You can see here the changes occurring at this third position. It is there, this one third position and this one is a first position. So first position losing a phosphate molecule, and that is nothing but forming a 3-phosphoglycerate. Now we will move towards the next reaction, reaction number 7. So in the reaction number 7, this 3-phosphoglycerate converted into 2-phosphoglycerate. 3-phosphoglycerate converted into 2-phosphoglycerate by phosphoglycerate mutase. By phosphoglycerate mutase, and this 3-phosphoglycerate mutase is a reversible reaction again. That is nothing but in the presence of phosphoglycerate mutase, and this occur as a 2-phosphoglycerate has been formed. This is one of the, we can say, isomerization reaction, in which what 3-phosphoglycerate convert into 2-phosphoglycerate in the presence of phosphoglycerate mutase, and this is nothing but we can say one of the reaction of the energy generation phase. This will change the OH, and this nothing but replaced by the phosphate molecule, and here the phosphate is replacing with the OH molecule, and that is nothing but forming a 2-phosphoglycerate. Now we will move towards the next reaction. In this reaction, what will happen? Phosphoenol pyruvate is generated from 2-phosphoglycerate. So 2-phosphoglycerate can convert it into phosphoenol pyruvate in the presence of enzyme enolase. So this one is a 2-phosphoglycerate. This 2-phosphoglycerate, again it is a reversible reaction, and there is nothing but the enzyme required is enolase, and this enzyme required magnesium or manganese and specifically O, and phosphoenol pyruvate is formed. This is again a high energy compound. So this 2-phosphoglycerate converted to phosphoenol pyruvate in the presence of enolase, reaction is reversible reaction. Now the last reaction of it, you can able to see here, this one is a nothing but the OH is replacing with a double bond formation, and that is nothing but forming a phosphoenol pyruvate. We are coming at the end of these reactions of glycolysis. So what will happen? The enzyme pyruvate kinase enzyme means what? It may be phosphorylation or it may be dephosphorylation. So it will transfer the high energy compound phosphoenol pyruvate. This phosphoenol pyruvate, it is an irreversible reaction. In the presence of pyruvate kinase, it is forming a pyruvate. So pyruvate having a two form. One is a enol form. Enol form means what? The second carbon is having a hydroxyl group and double bond is formed. That is nothing but double bond is already there in phosphoenol pyruvate. So pyruvate enol form means what? It is having a hydroxyl group. So in this, what will happen? This again a substrate level phosphorylation. It does not involve any ETC, electron transport chain mechanism, and that's why this reaction is forming from substrate directly the ADP converted into ATP in the presence of magnesium ion. So in this, what is changing? See here the phosphate molecule, and changing is nothing but a hydroxy molecule. So again, this is a second substrate level phosphorylation without involvement of the electron transport chain mechanism. So this is nothing but the irreversible type of reaction. So three irreversible reaction. First reaction, that is glucose converted into glucose 6 phosphate, and again next is fructose 6 phosphate converted into fructose 1,6 bis phosphate. The third irreversible reaction is phosphoenol pyruvate converted into pyruvate enol form. So enol form is hydroxyl group. Now we can see the keto form. See, same phosphoenol pyruvate in the presence of pyruvate kinase, it is forming a pyruvate keto form. Keto form will have the second is double bond carbon within the in between the oxygen and the carbon, and this will a last step of the reaction. So we can able to get two ATPs are used in energy investment phase. Then there are one NADH is formed. This NADH is nothing but we can say it is formed in the glyceraldehyde 3 phosphate to the 1,3 bis phosphoglycerate in that. So this ATC means what? 6 ATPs are formed, and the remaining is nothing but in a two reaction 2 ATPs are formed. So every time it is double. So 3 ATP NADH, 1 NADH is formed, 3 ATP. Because the two substrates are there, so 2 NADH is formed. So it is a 6 ATPs. Now in two reactions successive, 1 ATP is formed, but substrate is 2, so 2 ATP is formed. So total 10 ATP is formed in energy generation phase. Total 10 ATPs are formed, and investment is nothing but 2 ATP. So if we deduct, so it will form 8 ATPs from glycolysis. I am repeating, total we got 10 ATPs, investment is 2 ATPs. If we subtract, then it is forming a 8 ATPs in the reaction. So one glucose molecule oxidizes in a glycolysis to form a 8 ATPs. So we are having specifically in this reaction keto form, how it is formed, pyruvate, how it is formed. So this will nothing but we can say these are the various references I had used. And thank you very much for kind listen. And moreover, if you require any biochemistry's presentation or biochemistry topic how we have to discuss, please comment me in comment section, and subscribe for more new uh, videos on biochemistry as well as in pharmacognosy and phytochemistry. And here I want to give a disclosure that we are using these notes for the educational purpose for the students, and please don't take as a reference for treatment or element or other uses. Thank you very much. You can subscribe my channel for the more videos on biochemistry as well as on a pharmacognosy and phytochemistry. Thank you very much.